Thank you all for being here. This is a great honor to receive this award, but I'd like to thank ASM for 40 years now. My first scientific meeting was Binghamton in 1974. I've been coming to these meetings ever since because of the support, the interactions, the camaraderie of scientists here that we've all grown up. This is a big academic family. On this slide, I've added a bunch with the opportunity to make a Merriam address, you've got to find a topic. And I've talked about all these topics in recent years, and I know the worst Merriam address is a scientific chronology of things that you've touched on in your career. So I've taken a sort of unusual tack of actually repurposing my favorite lecture in a biogeography class I teach, which rather immodestly features research that I've been involved in, but it covers a very big topic, the evolution of South American mammals. This is a topic I've been involved in personally for 30 or 35 years. And it offers a very interesting juxtaposition because of the nature of the topic of paleontology and neontology and their respective limits and roles in informing us about the evolutionary history of faunas that I hope will, in the course of this talk, provide each of you with a new, if idiosyncratic, view of South American mammals and help explain what I've been up to over the years. As we saw yesterday in Jose's talk, South America is famous for having rich mammal faunas. About a quarter of the world's mammal species inhabit South America. It deserves our attention, respect, and understanding for that reason alone. But we also know that throughout the Cenozoic, South America was populated by creatures even more remarkable and amazing than those that populate it today. A few of those groups, archaic ungulates and cingulates and pilosans, are shown here. We all have the opportunity to learn a lot more about this episode of South American mammalogy with a book coming out in September by paleontologist Darren Croft, this Indiana University Press book, illustrated by Velizar Semyonovsky, that horned armadillos and rafting monkeys that recount some of the paleontological history. The major episodes in South American mammal evolution were laid out in a famous paper by George Gaylord Simpson called The History of the Fauna of Latin America. And as shown in this slide, Simpson broke this South American mammal history into three main episodes organized around strata in the South American fossil record. Stratum 1, the deepest, oldest stratum, was filled with things like marsupials and xenarthrans among the living fauna, as well as a variety of archaic ungulates, presumably derived from condylars. That was the earliest episode at the beginning of the Cenozoic. In the middle of the Cenozoic, two major groups show up de novo in the South American fossil record, the New World monkeys and caviomorph rodents. And it's actually only at the end of the Cenozoic, in a time period called the Great American Biotic Interchange, that many of the mammals that now seem absolutely emblematic of the South American fauna, like peccaries and tapirs, jaguars show up in the South American record. So these three episodes, although Simpson was a diehard opponent of continental drift, these three episodes actually identify South America's plate tectonic relationships throughout the Cenozoic. So these are Chris Scottizi's reconstruction maps of South America during the middle of the Mesozoic, when it was clearly part of Gondwana and shared a widespread Gondwanan fauna. By the late Cretaceous, South America was joined to Western North America by an ephemeral landbridge here. This is how marsupials moved from the northern hemisphere into South America. It's also presumably the colonization route by which condylars, which gave rise to some of the archaic ungulates, arrived in South America before its final separation from all other continents during the Cenozoic. we'll see its separation from Africa had already taken place by the Cretaceous Paleogene boundary, but it remained connected through Antarctica until the middle of the Cenozoic, and that's an important part of the story, as we'll see. So in terms of its continental separations, just to summarize those here, it was long separated from Africa by 100 million years ago, from North America by the KPG boundary, and from Antarctica about halfway through the Cenozoic. This is the molecular group of the mammal tree of life's reconstruction of living mammals. It shows the full array of mammalian diversity, living mammalian diversity here. And I just want to point out three things about this phylogeny. One is that living mammals have a very remote ancestry. Their common ancestor lived back in the Triassic. On the other hand, most of the modern radiations are quite young and date to the Cenozoic. And finally, that the Stratum 1 taxa that we're really interested in this talk, marsupials and xenarthrans, are very early branches that date back 100 million years ago and are presumably Gondwanan in age. And a developing paradigm within historical biogeography put forth cogently by Juan Morón is that the world's really divided into three main zoogeographic realms: a holarctic realm, a holotropical realm, and an austral realm, whose elements in South Africa, Australasia, and South America, including Antarctica, are clearly derivatives of Gondwanan age faunas. In South America, this is the distribution of the austral realm and its transition zone to the holotropical realm. And I had the very good fortune early on in my career, you can tell how long ago that was, to join this guy who's sitting in the audience, Peter Meserve, who was on sabbatical in Chile in 1984. I'd been on some coastal islands, and Peter invited me to join him at his study site on the slopes of Volcan Osorno. And in the course of 3 weeks, we put together this really remarkable collection that we wrote up subsequently in the Journal of Mammalogy. And I just want to call your attention to this line, in which we report 23 Rhyncolestes raffineris, a name many of you have probably never heard of, and I think 22 of them are lined up in this drawer. But at the time we made this collection, there were 4 specimens in the world's museum collections. Similarly, the second line, Dromesiops australis, another animal that most of you have probably never heard of, we had 61 captures of this, and it represented about 200% of what was known about these animals worldwide. Well, just to introduce them to you, since they both are members of this austral realm we're talking about, these are the shrew opossums, and this is an order of mammals entirely distributed in the Andes of South America. Here's Rhyncolestes from the southern Andes, a sister group, Les Toros from Machu Picchu and western Bolivia, and finally the northern shrew opossum up here in Colombia and Venezuela and Ecuador. Now, although this group is now restricted to the Andes of South America, throughout the Cenozoic, it had a much broader distribution from the Paleogene and Itaboraí and Brazil, clear up to Colombia and south to Patagonia. And if we look at not only its temporal distribution from the early Eocene onward and its phylogenetic relationships, the living Coenolestids form a derivative of a very generalized basal branch on this tree, and much of the diversity of the Paucituberculates are unfortunately no longer with us. However, among the living forms, we continue to discover new species of Coenolestes. There are only 5 species in this genus, but 2 of them have been described in the last 20 years, both in the Journal of Mammalogy, Coenolestes condorensis here and Coenolestes sangai, which was on the journal cover a few years back. Students working on the Coincides of the Colombian Andes, where the topography is great and the attention to specimens and collections has been historically low, but now is focused and continuing, will undoubtedly uncover additional species of this genus in the next few years. Now, the other austral marsupial we ran across at La Picada was the Monito del Monte, Dromesiops scleroides. And here's the entire distribution of this animal is here. Just in the last issue of the Journal of Mammalogy, Guillermo Delia and his colleagues subdivided this geographic range, making 3 species out of what had been 1. But Dromesiops is a very peculiar animal. This is another order of mammals represented only by this particular range. Dromesiops, I regret in a way I cannot even express, that the only evolutionary study that I did on this animal was published back in 1987 with Milton Gallardo, in which we described a very unusual chromosomal feature of these animals. It's called sex chromosome mosaicism, and what it involves is the loss of the Y chromosome in somatic cells of males. So males end up with a 13 chromosome karyotype, whereas females in somatic cells have this standard 2n = 14 karyotype that's primitive for marsupials. Now, this is unknown in other American marsupials, but is widespread among Australian lineages of marsupials. And at the time, we attributed this to an interesting parallelism, I think is what we called it in this paper, not realizing, as Sanger sequencing would show us in a few years, that this was really a synapomorphy uniting Dromesiops with the Australasian forms. That's shown very clearly in this paper, which shows the distribution of retroposon inserts over the 7 extant orders of marsupials. We've color-coded their names here. Here are the American lineages of marsupials, the 3 orders of American marsupials. Here are the 4 orders of Australian marsupials. And you can see that Dromesiops here, the Microbiotheria, shares many characteristics with the Australian marsupials to the exclusion of the other American groups. This sister taxon, sisterhood relationship between Microbiotheria and the Australasian forms, interesting as it is, took on new significance with the discovery in 1999 of Australia's oldest fossil marsupial in the Murgon Formation of Queensland about 55 million years ago. This animal was subsequently found to have a very interesting set of characteristics and to recast the relationship between Dromesiops and the Australasian marsupials in a new light. Fred Zille, back in 1982, in a study of postcranial anatomy of marsupials, identified ankle bones as one way to distinguish American marsupials from Dromesiops. And here you can see this ankle joint has 2 facets that are united in Dromesiops, and here is a single joint that's been subdivided in Dromesiops. And interestingly, this Murgon Formation form, Darthia, shows precisely this Australasian combination of characters. So its presence 55 million years ago in Australia suddenly cast the relationship between Dromesiops and the Australasian in new light. And we can see that here in this color-coded. Australia, again, is in red. South American forms are in blue. You can see Dromesiops here, sister to all of the living marsupials of South America, but outside that group is a 55 million year old Australian fossil. And here's another Australian fossil outside that. This relationship is not quite as solid as these, but at the very least, we have evidence, the suggestion that Dromesiops is actually derived from Australian marsupials and represents a back colonization of South America. And the continental connections, the Antarctica as a durable land bridge connecting Australia with southern South America provides a vehicle for why this relationship should hold true. Once we've established this linkage, other lines of evidence start falling into place. Rosendo Pascual described a 63 million year old platypus from Patagonia back in the early 90s, and a week or two ago in the journal Paleontology, a number of Argentinian authors have found diprotodont marsupials. These are relatives of kangaroos and wombats in the fossil record in Argentina. And so this South American-Australian connection via Antarctica appears to be a very important one in the distribution of some of these early taxa. Now, the second major stratum in South American fauna happened when South America was an island continent, much like Australia is today. It had no connections, save this one that lasted till about 31 million years ago with any other continent save Antarctica. And it was during this period that two diverse groups show up in the South American fossil record and quickly diversify. These are the caviomorph rodents and the platyrine monkeys. Simpson's conception of this stratum extended back to the Desiadin, which is about mid-Oligocene in age. But 10 years ago or so, John Flynn and his colleagues added the Tingarican fauna of Chile to this, which took the stratum 2 back to the beginning of the Oligocene. More recently, the Contamana faunas in Peru were discovered, taking this all the way back to early Middle Eocene. And you can see here's the earliest first appearance date of monkeys back at 26 million years. The prior date of caviomorph rodents, this is the Tingarican date, here at 32 million years, and extending it back now to 41 million years. These are things fossils can do for us. It's a pretty dynamic field. Just last year, Mariano Bond and his colleagues described a new species of monkey that dates back to 36 million years ago in Peru. And so stratum 2 starts earlier and is more extensive than we had previously thought based on these continuing fossil discoveries. Now, I had dabbled with stratum 2 fauna until the work of Nate Upham at the University of Chicago, and Nate's project taught me a lot about this group and its diversification. Nate chose to work on the Octodontoidea, and as shown in these graphs by colored portions, the octodontoid rodents comprise about 2/3 of the genera and 3/4 of the species of all caviomorphs. This is the distribution of the superfamily, and you can see from its distribution that it reaches its peak diversity in Atlantic forest Brazil and throughout the Amazon, but the group actually extends from Tierra del Fuego up through the Antilles. Now, this group had been the subject of many prior biogeographic analyses, but we used an analysis called Lagrange that uses modern distributions and the phylogenetic relationships of species today to infer the distributions of ancestral nodes in the tree. And you can see from the color codes here, these are all Patagonian taxa. Their ancestor is inferred to be Patagonian, so on back through the tree. And as you see, these green things here, which all correspond to Amazonian taxa, which are most generalized in terms of morphology and by Osvaldo Regg's hypothesis, were inferred to be the original forms of octodontoids that later diversified and colonized high latitudes, are rather robustly refuted by our analysis, which reconstructs most of these ancestral nodes as being high latitude. So this is another group that appears to have first diversified and become widespread at high latitudes, and only later in the terminal stages colonize the moist forest of Amazonia. Nate's dissertation largely involved extending this work, and over the course of his dissertation, he ended up producing, sampling every genus of hystricognath rodent, which is a vast extent. As you can see, each of these columns is a hystricognath rodent genus. The gray, this is the gene sampling up at the top. The gray squares represent sequences that could be downloaded from GenBank. The black sequences are sequences Nate recovered in 3 different labs in 3 different countries. And the white squares represent holes in the supermatrix we analyzed. But you can see that this tree comes up with very strongly reconstructed nodes for most. The hystricognaths are monophyletic. The caviomorphs here are monophyletic. They're sister to the phyomorph rodents of Africa, and outside this group are the old world porcupines that have been problematic for many years, and so on. We could talk more about the topology at another time, but what I want to call your attention to is all the yellow squares on this, and those yellow squares represent fossil calibration points. One of the topics that first attracted Nate to this group was the vast diversity of fossil caviomorphs and the opportunity for using internal dating calibration points to date nodes and hence be able to accurately infer evolutionary rates. The group shows enormous variation in karyology and body size, et cetera, that can be analyzed with this kind of data. With those calibration points, we could provide dates. So here's the split between caviomorphs and phyomorph rodents at 42 million years. The appearance of the 4 superfamilies of hystricognath, of caviomorph rodents, octodontoids sister to chinchilloids, and cavioids sister to erythizontoids. One curious feature that appeared out of this tree in a manner that took us all by surprise was this very long branch leading to a genus of phyomorph rodent. This is a branch that appears at the same time as the branches subtending the superfamilies of caviomorphs, and it leads to a genus of African rodent. And when we look at this on a histogram sort of basis, we see that on our previous tree, most of the genera that appear on that tree originated in the middle to late Miocene, with a few stragglers into the Pliocene, but nothing back here in the Oligocene when most of the families and superfamilies were appearing. This one genus is Heterocephalus. Heterocephalus is the naked mole rat. And in a paper we published a couple of years ago, looking through naked mole rats and inferring that they'd been an independently evolving lineage for 31 million years. A lineage like that will undoubtedly accumulate lots of audapomorphies, and the list of audapomorphies for this family is absolutely enormous. The Methuselah mouse, it's resistant to tumors, it tolerates hypoxia levels that lead to brain death and many other forms, and so on. So that led to an article arguing for its elevation as to family status. Finally, moving on to Stratum 3, this is the latest episode in South American mammals. These are deposits that include most of the groups that appear there, and paleontologists historically have completely ignored most of neotropical diversity because they left out of the picture mice and bats, and of course those are 2 enormously diverse groups in the neotropical fauna. But when we break the South American fauna, neotropical fauna, into the strata, you can see that Stratum 3 actually includes most of the fauna and is a very important period for us to understand. Now, fossils have told us a lot about the Great American Interchange, Biotic Interchange, that's what GABI stands for. In its early phases, we thought of 3 million years as a closing of the gate and when all of this could take place. But we know from earlier appearances of South American taxa north of the Isthmus of Panama and of North American taxa south of the Isthmus of Panama that this was actually taking place over a much broader segment of time. And we are now sort of inclined to postulate, as Mike Woodburn did, an island arc connecting these 6 million years before present, and it's now going much earlier than that. In fact, just earlier this year, Panama cebus was described a monkey 21 million years ago in North America. And so the Great American Biotic Interchange has gotten a lot more fuzzy, and the dates on it become a lot more diffuse than anything that we expected on the basis of classic literature. A very nice way to look at that using modern faunas is through a dated chronology of these different groups. And Eduardo Aziric did this in a paper looking at the phylogenies, the chronograms of 5 families of neotropical carnivores. The dates of the closure of the isthmus and the formation of the island arc tells us that for some groups, like the foxes, clearly the foxes diversified after the Great American Interchange and probably in situ in South America, whereas for groups like the raccoon family or the weasels, clearly a lot of those divergences had to take place long before the isthmus was closed and South America colonized. This kind of analysis using the phylogenies of modern taxa to infer diversification timing and rates was carried by Julio Villela to the most diverse group of South American mammals, and these are the sigmodontine rodents. 84 genera, almost 400 species, and you can see from this slide, which includes the same marker, that most of the sigmodontine diversification took place long before the postulated closure of the isthmus, 10 to 12 million years before present. So either Central America was a very crowded staging area for this, or these guys got into South America long before the postulated age. One thing we can say for sure is that the colonization of South America by sigmodontines has produced a very different pattern of diversification than had developed over 40 million years among the caviomorph rodents. And here's what the present-day diversity patterns of sigmodontines look like in South America. Here's one of caviomorphs, and you can see that they respond rather differently to the same landscape and climatic scheme. This is work done by Renan Maestri in my lab. I want to sort of close on this topic with a study done by Paul Velasco. Paul was a student in my lab a decade ago. He's now a postdoc at the American Museum. He worked on the genus Platyrrhinus, but during his final summer in Chicago, we worked together on a topic first introduced to me by Victor Pacheco. We studied the genus Sternira. This is, as you can see from the distribution of these type localities, quite a diverse genus. It's widespread. It shows a neotropical diversity, neotropical distribution from one end to the other. And we were able to, through the help of lots of colleagues, many of you in the audience, we were able to assemble genetic samples of Sternira from throughout its range, many of these localities represented by 2 or 3 species of Sternira, and create a phylogeny from this. Sternira, the 21 reciprocally monophyletic lineages on this, the 3 shown here in red, represented species without names. The ones we named after Burton Lim and Robert Baker in a 2014 article, and we elevated various subspecies to specific rank. So Sternira is a lot more complicated than it was before, and it's a long way from being fully resolved. But this is the biogeographic analysis of Sternira, and it's very interesting with its implications for the Great American Biotic Interchange. Sternira, of course, is a bat. It can fly. And unlike many other groups of South American bats, it's found on a bunch of Lesser Antillean islands. And so we all thought, even Carl Koopman thought, that Sternira was a good overwater colonist, and it would have easily gotten into Latin America, Central America, long before the interchange. Instead, when we do this analysis of Sternira, using this template here, and keep your eyes on the blues and greens, which correspond to the northern and central Andes, and the greens, especially this lime green, which is Central America. Over here, you can see that all these basal nodes here are light blue and dark blue. And that says the first 3 branches on this tree are all species limited to the Andes. This group obviously originated in the Andes and began its initial diversification in the Andes. It moved into the Chocó, this area on the Pacific slope of Colombia. And the first trees, nodes in which it appears in Central America are actually pretty shallow. They date to the Great American Biotic Interchange. What's especially interesting to us is that where these nodes leading to red, which is the color of the Lesser Antillean populations, are all quaternary in age. This is a time period in which sea level lowering led to the extent, the retraction of oceans from the sand arcs that connect the Lesser Antilles that would have greatly shortened the inter-island distance between these islands and made their overwater colonization by Sternira much easier than it would be at any point during earlier periods of time. So Sternira was behaving much like gomphotheres were when it came to Great American Interchange dynamics. Finally, I want to end this talk with, we talked about episodes of diversification, to talk about episodes of species discovery. This is a map worldwide put together by Deanne Reeder and her colleagues on dates of description of mammals worldwide. And you can see that this graph shows a steady increase, peaking in the late 19th century and early 20th century, and then falling off as biologists embrace the polytypic species concept and the modern synthesis here in the middle of the 20th century. But on the other side of this, we see a steady increase. And this sort of new age of discovery is something that systematists across biological fields all recognize and feel we're involved in. On the right side is another interesting interpretation of this, because it looks at where these authors were writing from. And across the bottom are literature as a source of the specimens, but then whether the authors were writing from continental Europe, largely Germany and France, or the United Kingdom, largely Oldfield Thomas in his office at the British Museum. Over here is, in the 4th column, is North America, which was booming in the days of the biological surveys of the Smithsonian. And finally, this last column is South America, and you can see that in the final decade, the final half century, South American authors were contributing almost as much to the description of South American faunas as North American authors were, and far outdistanced the rest. And clearly, if we redid this analysis in the 21st century, we would find this dominated by Latin American authors. A very happy outcome. I just wanted to say the drivers of this internationalization are clear. One is open electronic access to critical resources, and this library resources as well as collection resources through online access. There's been lots of programs for international support, including programs like our own Latin American Student Research Fund and the Pearson Fund. But beyond this, there are now national fellowships for studies abroad, and in the 80s we all were beneficiaries of students visiting from Chile and Mexico and Venezuela and many other countries. More recently, Brazil and Argentina have been especially active in sending their students out so that they come back with international training and connections and can carry this forward. Ecuador introduced a program for national support for visiting professorships to infuse new life in their educational programs. But all of this has led to expanded access to advanced scholarship throughout Latin America. I myself, at the moment, am the beneficiary of these programs. I have students from Argentina, Brazil, and Colombia, all presenting papers at this meeting. Julio Anderson and Natalia are all presenting papers in poster sessions tomorrow and Tuesday. I hope it's their first ASM meetings. I hope you all drop by and say hi. But finally, let me summarize here by saying Simpson's 3 strata continue to be useful for conceptualizing faunal stages in South America. They all started earlier than anyone had believed in the past. They all have fuzzier boundaries, but they still represent important, useful concepts for subdividing the South American fossil record. The fossil and recent studies offer incomplete but strongly complementary views on the development of this. We're still very much in an age of discovery, and systematics continue to provide a foundation for studies in all other branches of science. Many of us have deplored the decline of natural history in North America, but I can assure you it's alive and well in Latin America, and anyone who's attended mammal meetings in Brazil or Argentina knows that the rooms are full, they're thronging with young students, and it's something that we should now feel challenged ourselves to inspire our own students to a similar extent. And finally, it must be obvious from the attributions on the work I've presented today that this talk would be immeasurably poorer without the collaborations of a host of students and other colleagues. So some here today. Thanks very much. Thanks.
We do have time for some questions.
Bruce, when marsupials were moving back and forth, or at least one direction between South America and Australia via Antarctica, Africa was right there next to them, wasn't it? But can you offer an explanation for why there's not evidence of marsupials?
The question was if Antarctica was a durable land bridge throughout the Paleogene, where was Africa in this, and why wasn't Africa similarly colonized? And Africa was out of the picture when it came to this land bridge by 100 million years ago. So it was long out of the picture, and that, I guess that's kind of a confusing map I spliced in there from the American Journal of Botany. So you see there are different dates for each of these connections, and the one between eastern Antarctica and South Africa is 165 million years. So it's kind of a single image, but the arrows on it indicate different chronologies of